Hello, everyone. Welcome to today's live broadcast, Enhancing Checkpoint Blockade in Lymphoma with Situ Vaccination. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. Today's webcast is presented by LabRoots.com, the leading social media site for science professionals, and sponsored by Fluidigm. Fluidigm develops, manufactures, and markets life science analytical and preparatory systems for markets such as mass cytometry, high-throughput genomics, and single-cell genomics. We sell to leading academic institutions, clinical research laboratories, and pharmaceutical, biotechnology, and agricultural biotechnology companies worldwide. Our systems are based on proprietary microfluidics and multi-parameter mass cytometry technology and are designed to significantly simplify experimental workflow, increase throughput, and reduce costs while providing excellent data quality. Fluidigm products are provided for research use only, not for use in diagnostic procedures. To learn more about Fluidigm, please visit www.fluidigm.com. Before we start, there are a few instructions. We want to hear from you during this interactive broadcast, so please ask questions or leave us a comment. Answers welcome too. And you can do this by hitting the green Q&A button at the lower left of the presentation window and typing in your comments and questions. We'll try to get to as many as we can, and we'll follow up if we don't have time today. Want a better look? You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see this presentation properly, let us know by clicking on the support button on the top right or use the Q&A button. We'll make sure we try to resolve any issues. Now let's get right to today's presenters. We are proud to welcome Michelle Poulin, PhD, and Joshua Brody, MD. Dr. Poulin earned her bachelor's degree from Smith College and her PhD in immunology from the University of Colorado, Denver. After completing a postdoctoral fellowship at National Jewish Health in 2003, she joined BD Biosciences as an instructor in the Customer Education Department. In 2012, Dr. Poulin joined DVS Sciences as a field applications scientist and became manager of the North American Field Applications Scientist team shortly after DVS was acquired by Fluidigm in 2014. Dr. Brody is an assistant professor in hematology and medical oncology and the director of the Lymphoma Immunotherapy program at the Icahn School of Medicine at Mount Sinai in New York City. He is also a member of the Lymphoma Research Foundation's Mantle Cell Lymphoma Consortium and a Lymphoma Research Foundation Career Development Award grant recipient. Dr. Brody received his MD from State University of New York Stony Brook School of Medicine and his bachelor's degree in molecular and cellular biology from Harvard University. He completed his residency in internal medicine at Yale New Haven Hospital and his fellowship in medical oncology at Stanford University School of Medicine. With clinical focus in chronic lymphoid leukemia, cutaneous lymphomas, follicular lymphoma, mantle cell lymphoma, and post-transplant lymphoproliferative disorders, his lab at Mount Sinai focuses on basic and applied tumor immunology. I will now turn it over to Dr. Poulin, who will be giving a brief overview of the mass cytometry technology before Dr. Brody presents Enhancing Checkpoint Blockade in Lymphoma with Situ Vaccination.
Thank you, Judy. As Judy just mentioned, I just want to give you a short introduction to the mass cytometry technology. As you're all well aware, biology is inherently heterogeneous, and biological systems consist of heterogeneous cell types, each of these with diverse functions and functional states. Such complexity demands the use of high-dimensional proteomics panels that can simultaneously measure breadth and depth in your system of interest with single-cell resolution. Mass cytometry uniquely enables this with the ability to look at more than 50 parameters simultaneously on millions of cells. It uniquely enables both the discovery of new biology and comprehensive functional profiling across a range of research areas, including basic research, drug discovery, and clinical research. To describe mass cytometry technology, I'll explain the 5 elements of mass cytometry. Firstly is metals. We use metals as our tags, and we have more than 50 high-purity metal isotopic tags to be used. Because of their high purity, we see minimal background from signal overlap between these tags, and because these metals do not occur naturally in biological systems, we do not get background from endogenous cellular components. Then we build panels. We can build large dimensional panels using our MaxPAR metal conjugated antibodies. There are more than 600 pre-conjugated antibodies in our catalog, which enable both phenotypic and functional applications. These antibodies can be purchased individually or as part of pre-optimized panel kits. In addition, we sell labeling kits in 35 different metals that allow you to conjugate your metal of choice to your antibody of interest, and Fluidigm also offers custom conjugation services. In addition, we offer cell ID reagents, which are non-antibody based, such as barcoding, intercalators, which identify single cell events, and cisplatin as a dead cell indicator. With these high dimensional panels, we then stain our cells of interest with a method very similar to what you may already be using for flow cytometry. These stained cells are then passed in a single cell suspension into the Helios instrument, where these cells are ionized in our high energy plasma into an ion cloud. From there, the ion cloud is transmitted into our detection chamber, where a single detector is able to read and identify each of these separate tags with 1 Dalton resolution. Our third generation mass cytometer, the Helios, has 135 mass channels from 75 to 209 Daltons, which allows us to measure all of our existing tags as well as those that are developed in the future. And lastly, we come to cells. The Helios collects multiparametric data for large numbers of cells per sample, enabling proteomic profiling for a breadth of cell types, all in a single tube. And just as a way to conclude, I wanted to show you a graph showing the ramp of publications utilizing mass cytometry. And as you'll see, over the last few years, there has been a very large ramp in these papers showing that the technology is becoming widely accepted, and many new researchers are publishing their research that has utilized this breakthrough technology. And I'll hand it now back over to Judy.
Thank you, Dr. Poulin, for that informative presentation. I will now turn it over to Dr. Brody.
Judy, thank you so much, and Michelle, thank both of you guys for the introduction and the explanation, and thank you to everyone who's joining throughout the country, throughout the world. I'm going to take a little time to tell you some application of this very advanced form of immune monitoring that we've incorporated into a lot of our clinical trials. This first slide is just our aspiration and a little bit of our success. We've actually had patients doing well with novel immune therapies that we've developed, and a lot of that is due to our ability to understand how these immunotherapies are working. Our goal here is to learn how to improve immunotherapies for cancer overall. Certainly the most famous subclass of immunotherapies now we refer to as checkpoint blockade. These are perhaps the greatest breakthrough in cancer therapy in the last 4 decades, but still with a huge amount of room for improvement. These therapies help a lot of people, but ultimately the majority of cancer patients still don't get benefit. We need to improve checkpoint blockade, and this approach we refer to as in situ vaccination has been our focus. This is our institution, Mount Sinai School of Medicine in New York, and this is the concept of in situ vaccination. We started some of this work when I was working at Stanford in California, the idea that we can make a vaccine not just in a factory as we do with flu vaccine and hepatitis B vaccine. All of these vaccines are conceptually a similar thing. They are just bringing together something we want the immune system to hate, something that is an antigen and hopefully an immunogen, and somehow co-localizing that with something which is immune stimulatory. And some vaccines co-localize that thing automatically. A live flu vaccine has influenza virus particles which are naturally immune stimulatory. But we don't have to co-localize the antigen and the immune stimulant in a factory. We can do it right here at the tumor, and that was the idea here in these first trials. We have these patients with lymphoma, all of these B-cell derived lymphomas, this common cancer that we see every day in the clinic. All of these lymphomas express toll-like receptor 9. Therefore, if we co-localize a TLR9 agonist, CpG, we can convert those tumor cells into antigen-presenting cells. They can present some of their own antigens. And these are not professional APCs, but they can still present antigens to a degree. These tumors also have professional APCs within them, dendritic cells. Unfortunately, they have very few. But hopefully we could activate some of those professional APCs as well with co-localized TLR agonists. And between these two amateur and professional APCs, hopefully we could activate some tumor-reactive T cells. Those T cells would show that they're activated by expressing surface markers like CD137, by secreting anti-tumor cytokines like interferon gamma, and possibly those T cells might be doing 2,000 other things that we haven't shown you on this picture, and ultimately that's going to be largely the purpose of why we're including this discussion in the context of mass cytometry, because we don't know really what to look at on that T cell. I mentioned two things here, but there could be many, many other markers, cytokines, or combinations thereof, and that's why we need to be able to look very broadly at those potentially tumor-reactive T cells. But this was the concept as we began the in situ vaccine approach. We brought this concept into a clinical trial. We treat patients at one site, one tumor, with something called mini-dose radiation, and then there's intratumoral injections of this TLR agonist called CpG. And we don't look if that site regresses or shrinks. That would really be cheating, because the radiation by itself might just cause regression. But if we see tumors that are far away, that were not irradiated and not injected, if we see those tumors shrinking, for us that is a real indication of inducing anti-tumor immunity systemically. So that is really our readout in these trials. So the question is, can we induce systemic regressions of these patients' tumors? And the answer is that we can. This was the first 15 patients that we treated. We treated a total of 60 patients, and as you see on the right here, some of these patients had partial or complete regressions of their tumors, and again, at sites far away from the site that we injected with the immune stimulant. So that was extremely gratifying for us. We published this now already a few years ago, and this is just one picture of one of those patients. You see on the top left, I'm not a radiologist. Most people on the call are not radiologists, but in radiology, a great hint is if you see a green circle, that's usually a bad thing. And you see this green circle around this bulky tumor in the back of the person's abdomen, kidneys on the left and right, liver over here on the left, backbone at the bottom, belly button at the top, and this green circle is a bulky tumor. We see that this patient was one of our first patients on that trial I described. They were injected at a site far away from this tumor, a site near an inguinal lymph node. And after therapy, you see this tumor now is a small green circle, so almost completely regressed. And alongside this tumor regression, we saw the induction of anti-tumor T cells. I won't go into this simple flow cytometry assay in great detail, but we basically co-culture the person's peripheral blood T cells with a sample of their own tumor that we obtained before therapy, and we see post-vaccination the person's CD8 T cells make more interferon gamma in response to their own tumor. So these are literally tumor-reactive CD8 T cells. And on the right side, we see that these CD8 T cells, specifically in the CD45RO high group, the memory CD8 T cell subgroup, express more CD137 when they see autologous tumor in vitro. So tumor-reactive T cells correlating with this bulky tumor melting away, very gratifying for us, very good for the patient in this case, because some of these remissions are ongoing for years. But the whole purpose here of stimulating the immune system at the site of the tumor is to accomplish what I'm going to call a magical thing, this thing called cross-presentation. So normally if tumor antigens are outside of dendritic cells or other APCs, those antigens, and this is all, I guess back to immunology 101, it should not be news to most of you here today. These exogenous antigens get endocytosed, and they very naturally go in through endocytic pathways that end up in them getting represented on class 2 MHC molecules and presented to CD4 T cells. That is good and important, but ultimately not what we think we really need in tumor immunotherapy. We think we need to educate CD8 tumor-reactive T cells. So that requires that these originally exogenous antigens somehow, and here's the magical part, magically get out of these endosomes or other vesicles, get into the cytosol, and now reprocessed through cytosolic pathways through TAP and then into MHC class 1 grooves so they can be presented on the surface to CD8 T cells, and we can induce anti-tumor CD8-mediated immunity. That whole trick, this cytosolic diversion of endocytosed antigen, is called cross-presentation, and we need to accomplish that here. We think that there are certain APCs that are able to induce cross-presentation, and foremost amongst those are dendritic cells. I mentioned this problem in the old paradigm is that we had a paucity of dendritic cells in the tumor. When we started those clinical trials, there wasn't much we could do about that, but now there is something we can do. We have access to clinical-grade FLT3 ligand. FLT3 ligand is the primary growth and differentiation factor of dendritic cell precursors. Sometimes we call it the erythropoietin of dendritic cells, or the GCSF nucleogen of dendritic cells. It can induce differentiation and eventually proliferation of dendritic cells. And now we can bring many dendritic cells into the tumor. Once they have endocytosed some tumor antigen, we can activate those dendritic cells with different TLR agonists. What I'll mostly talk about today is a TLR3 agonist, but we have probably a number of options there of which TLR agonist to use, as these dendritic cells do express a number of different toll-like receptors. So now after TLR agonism, we have a bunch of tumor antigen-loaded, professional activated APCs, dendritic cells, and hopefully these can really powerfully induce activation of tumor-reactive T cells, and as they get activated, they will proliferate and go induce systemic anti-tumor immunity. So that is the concept. We first did some brief assessment of how this actually can play out in preclinical modeling. In some of these animal models, we use tumors that are labeled with a fluoroprotein, M-cherry, so we can keep track of both the tumor cells, but also any antigen-presenting cells that ingest tumor cells because they become somewhat M-cherry labeled. I'll go briefly through this somewhat complex flow cytometric gating strategy we use to focus on different dendritic cell subsets. I won't go into detail here for the sake of time, and at the end of the day emphasize first just that we can increase the number of dendritic cells in the tumor, this first histogram here, using FLT3 ligand intratumorally. In fact, it works even a bit better when we administer the FLT3 ligand into the tumor as opposed to nearby, but really either of them work. The punchline really here is that FLT3 ligand can induce both systemic and intratumoral dendritic cell infiltration, both in the tumor and in the tumor-draining lymph node, as we see here. And looking at one of our earlier mass cytometric CyTOF experiments to make this point even a bit more elegantly, we're comparing here intratumoral FLT3 ligand on the right as opposed to intratumoral GM-CSF, another myeloid stimulating cytokine that historically has been used in really the vast majority of tumor immunotherapy vaccine-based trials. So GM-CSF is really the historical precedent, and now here we see with FLT3 ligand, this is a spade plot of mass cytometric data, so we are looking at really almost the entire immune repertoire throughout this tumor using a 34-plex mass cytometry assay. And then we are gating on many different subsets of dendritic cells. We've named them here. You can see on the right PDC, plasmacytoid dendritic cells, and all the way on the right different subsets of myeloid dendritic cells, CD11B dendritic cells, TLR3 expressing dendritic cells. And the color coding here is fold increase. It's actually a log fold increase as compared to the control, which was a saline control. The punchline of the whole experiment is that you see FLT3 induces many fold increase. I think these were up to beyond order of magnitude increases in TLR3 expressing dendritic cells and also CD11B dendritic cells as compared to controls. And overall, GM-CSF had a much milder effect on the intratumoral immune repertoire, especially on dendritic cell subsets. So we think FLT3L is very potent at bringing dendritic cells into the tumor, and hopefully dendritic cells that we think are good at cross-presentation because they've been described that way, and also some actionable observations about these dendritic cells that they express TLR3, and we should be able to activate them with TLR3 agonist. Just showing some of that same data again, flow cytometry, that the cells recruited by FLT3 are mostly TLR3 expressing, but we also have some CD11b dendritic cells as well. And when we look at what that looks like in the tumor prior to therapy, these tumors are extremely bland. They are non-inflamed. We sometimes call them cold tumors because they are not inflamed, and there's very little immune infiltrate at all. When we look at CD11c and TLR3 expression after FLT3 administration, we see massive infiltration of dendritic cells into these tumors. These red areas are CD11c expressing dendritic cells, and the green overlaid with red are the TLR3 positive CD11c dendritic cells. So we can bring dendritic cells of these subtypes into the tumor. They don't rest around the tumor periphery. They infiltrate well into the tumor. And we look even more at some other surface markers to more rigorously tell the phenotype of these dendritic cells. Here the x-axis are other markers of cross-presenting dendritic cells, XCR1, Langerin, CLEC9A. The z-axis, the color coming out at you, is the fact that most of these are highly expressed CD8 as well, and more of these express those markers than 11b. So same data shown in a few different ways, all getting towards the purpose of proving that these dendritic cells we recruit should be able to cross-present tumor antigens. One marker I didn't mention to you is CD103. CD103 has been a big focus both in our group and in the group of one of my mentors, Professor Miriam Murad, and her group, as well as a few others, have published the importance of CD103 dendritic cells in tumor immunotherapy and other contexts. This was a paper that we published in Immunity last year, showing here on the left that CD103 migratory dendritic cells were really uniquely able to cross-present tumor antigens. What you see here is a CFSE plot of anti-tumor T cells, just OT1 anti-OVA T cells really proliferating, but only when they are exposed to tumor antigen in the context of 103 positive migratory dendritic cells. And then just going all the way over here to this tumor growth curve on the right, this was, to me, amazing and even a little bit surprising story, but it has convinced me now that this most famous therapy that we use in cancer immunotherapy, PD-L1 blockade, we see gives some benefit. The black curve here is tumors growing in the unmanipulated state. The lower royal blue curve is the benefit of anti-PD-L1 antibodies, showing that those tumors grow more slowly, and some mice actually have tumor regressions. But the surprise here is that the light blue curve shows that that benefit of checkpoint blockade is completely lost when we redo the experiment in a dendritic cell depleted mouse, a BATF3 knockout mouse, which doesn't create these CD103 cross-presenting dendritic cells. So that's amazing to me. We think of checkpoint blockade as a T-cell therapy, and it is one, but that T-cell therapy relies completely on the a priori cross-presentation of tumor antigens by these dendritic cells. So if you want checkpoint blockade to work well, we do need dendritic cells, and what I'll try to convince you today is that perhaps the more dendritic cells, the better, as long as we're getting the right type of dendritic cells. I didn't show you CD103 dendritic cells in that mouse assay I described, and this is some of that same mouse data I already showed you, showing we recruit many TLR3 dendritic cells here on the right of this 3D plot, a smaller number of CD11b dendritic cells, but in the z-axis we see CD103, and there are really very few CD103 positive dendritic cells. As I mentioned, and when we translate this into our patient clinical trials, we include irradiation. So this is the non-irradiated control group, but then we were, I guess, gratified to see that when we add irradiation, 9-gray radiotherapy to that tumor site, many of these TLR3 positive dendritic cells are now expressing CD103. You know, in our belief, and I think we're probably right about this, is that these were just, we call CD103 precursor dendritic cells. As we see, they were the same in every other marker, TLR3, XCR1, etc., but only their final activation, and in this case that activation occurs after local radiotherapy, presumably releasing some damps, causes their expression of CD103. So at the end of radiotherapy, we do have these CD103, TLR3 positive dendritic cells. Hopefully they really will be able to cross-present tumor antigens to anti-tumor T cells. I'll just very quickly go through an in vitro cross-presentation assay that we use to assess this. We want to only study cross-presentation, not direct presentation by a tumor cell. So we have CRISPR gene-edited out beta-2 microglobulin from these GFP-expressing A20 lymphoma cells here. So they cannot present themselves. They don't have beta-2 microglobulin. They don't have class 1 on their surface, but they are really just a tumor source of antigen. And the antigen here is going to be GFP, because at the end of this assay we'll use a very cool readout developed by another lab neighbor of ours, Dr. Brian Brown, these Jedi T cells that you see on top, which are GFP-reactive CD8 T cells. So they will react hopefully to the GFP that's starting in these tumor cells. So we have GFP-containing class 1 non-expressing tumor cells. We co-culture them with dendritic cells that either have or haven't received TLRa agonist, and then we assess whether those hopefully tumor antigen-loaded dendritic cells can now cross-present to these Jedi T cells. We read out here, letter C, cross-presentation by those Jedi T cells proliferating, and then in D we read out whether these Jedi T cells can also kill. So just looking at each part of that, this is sort of the first part. We see these CD11C dendritic cells over 2 hours, 8 hours into the day, starting to eat GFP and becoming GFP positive. So that's sort of the first Petri dish you see here in the assay. The second Petri dish is now we see that we're looking here at the Jedi T cells, and they start to proliferate. Just moving to the bottom right of this assay, we see they proliferate best when the feeding antigen-presenting cells came from a mouse with FLT3L, and when those dendritic cells here on the bottom right were exposed to TLR agonist poly-IC. So we see 28.8% of these Jedi T cells proliferating only when the APC was the optimal APC in terms of FLT3L mobilization and poly-IC activation. So we can load these dendritic cells with tumor antigen, we can cross-present them to CD8 T cells, and then those T cells can go on to kill target cells. So the last part, this third Petri dish is now co-culturing those activated Jedi T cells with the normal tumor cell that does express class 1 and does express the GFP antigen. And just to summarize some of what's a much larger assay, when we don't add the Jedi T cells on the bottom row here, we see that we have mixtures of green and red lymphoma cells. And when we do add the Jedi T cells here on the top row, we see that those green cells are eliminated almost completely. It's gone down from 50% to 0.7%, or if we've activated the cross-priming dendritic cells with poly-IC, almost completely gone, these tumor cells down to 0.3%. And of course it only works when those cross-priming source of antigen actually had the GFP antigen. Here we see a control in the right column when there was no GFP antigen in the cross-priming. And then just one more version of this same experiment. We've enriched for a new type of lymphoma cell which is naturally more T-cell resistant and repeated the assay. And basically the outcome here is looking all the way to the right, we see that only when we have poly-IC activated dendritic cells do they effectively cross-prime. Now these sensitive tumor cells have gone down to 18% instead of the 62% now that they started with. So even with fairly resistant tumor cells, these cross-primed Jedi CD8 T cells can kill cancer pretty well because of the effective cross-priming they got from dendritic cells. So just to summarize the purpose of all of this, we basically reproduce what we do in clinical trials with the mice, inject FLT3 ligand into one tumor. Again, I already showed you that dendritic cells are recruited to that tumor. We irradiate that tumor, and when we do so, we see here on the right that many more of these dendritic cells are now absorbing the fluoroprotein tumor antigen, cherry, much more so after 9-gray irradiation than when there's no irradiation. And now when we activate these tumor antigen-loaded dendritic cells, these 9-gray, 8.9% and 9.8% dendritic cells, when we activate them with poly-IC at the tumor site, we get effective anti-tumor immunity. Here we see in the red curve the triplet therapy, FLT3, radiotherapy, and poly-IC is even much better than the doublet therapy. We don't even actually have the control group on here, which is the untreated mice. Their tumors grow very quickly, and those mice are usually dead by day 16. So a lot of benefit here at the tumor site, and also benefit on the right side of the page here at a distant untreated tumor site as well. And of course, the purpose of this is to understand the mechanisms enough that we can bring this to patients, and that's what we've done. We wrote an IND, got the FDA's permission, brought this into a clinical trial for patients where we basically do a newer version of what I described before. I'm always supposed to remember that we were very lucky to get funding from the Damon Runyon Cancer Research Foundation to allow us to do this trial. And basically the model is similar to what I showed you before. We inject FLT3L into a tumor, mini-dose radiotherapy, intratumoral TLRa agonist poly-IC, and again reading out regressions at distant tumor sites. So this trial is still ongoing, but we've treated a number of patients now. In those first few patients, we do some real-time immune monitoring. So these are fine needle aspirates of the patient's tumor that we're treating, and we see after FLT3 ligand therapy, these CD1C dendritic cells have now increased from 0% up to 6%. The 0% was really something like 0.01 or something, so it was about a 600-fold increase in these dendritic cells in the tumor, as we see in the middle panel here. Those dendritic cells don't stay there forever. We think they eventually traffic away to secondary lymphoid tissue, so we see they are mostly gone afterwards. We see a recruitment of CD141 dendritic cells on top here in the middle panel as well. We can recruit dendritic cells with FLT3 ligand, and then with radiotherapy we can activate those dendritic cells. So we see this blue gate means myeloid cells that are CD80 high and PD-L1 low, so we think of them as activated. And we see here going from before therapy, pre-treatment, to after myeloid cells in the tumor. And I think we may be paused now. And we see that those activated myeloid cells are in the tumor. We want to inflame the tumor, make activation of myeloid cells, but we don't want to inflame the entire person. And that's what we see, that if we look in the person's blood at the same time, whereas on the right side here the myeloid cells are activated in the tumor, you see on the bottom right the myeloid cells are generally not activated in the peripheral blood. So we hopefully are inducing tumoral inflammation, but not systemic inflammation. That is our goal, so it was gratifying to see even in these first few patients that we treated. And I will just quickly mention here that the black lines are the first couple of patients' overall tumor regression, and you see these patients are having 50%, 40 to 50% tumor regressions. The blue and pink lines are showing those same patients' red blood cells and platelets improving. But let me just show you a more beautiful picture conveying the same idea. On the left side here, we see one of the first patients, and this upper leftmost CAT scan is a coronal slice, meaning we are slicing the patient up and down. We don't actually slice the patient at all. This is just how the CAT scan reconstructs the picture. And we see again this ominous green circle sign. So you see on the right half of this person's neck, there's a big carotid artery and big jugular vein, as we should all have. But on the left side of this person's neck, you don't see an artery or a vein, because this giant tumor that we have circled in green is obstructing that vasculature, the artery and the vein. If you just move to the second picture there, it's the same thing. Now we're looking at the neck, but front to back instead of up and down. The chin is on the front, is on the top here. And again, you see with the green crosshatches, this same big tumor in the patient's neck. Really, you can see it sort of budding out from the patient's cheek and face. And this patient, when he walks in the room, he had a big tumor sticking out of the side of his face. But even more scary, you see it squishing the vasculature here. On the right side of his neck, you see open circles, white vasculature, blood vessels that are opened up. And on the left side, you see these squished, closed white vasculature. So bulky tumor in the patient's neck, and on the bottom panel here, bulky tumors in the patient's pelvis. These are called external iliac lymph node tumors, and they are, several inches each of them. So, small apricot-sized tumors here in the patient's pelvis. So he had advanced stage disease. We were treating this patient not at any of those tumor sites, but at a site that was also a left inguinal site far away from these tumors. And we see after the vaccination, now this person has blood vessels on both sides of his neck, And you see that giant green circle has become a small tumor with a little green circle around it. And on the transverse imaging, the far right upper picture, that we have a small crosshatch near and almost completely opened up vasculature with these big open white circles on both sides of his neck now. And on the bottom right, we see these bulky pelvic tumors almost completely regressed. So that was very gratifying, of course, for us, for the patient, for his family as well. This is showing you more data from that same patient, looking in the patient's blood. Flow cytometrically, we see that this patient's blood was actually full of tumors B cells. Again, these lymphomas are derived from B cells. They express B cell markers like CD19. And hopefully all of us, hopefully here today, have a healthy blood in which we have more CD5 T cells than we have B cells in our blood. But this patient is just the opposite. He has many CD19 B cells in his blood. They are almost all his cancerous B cells. And again, we didn't inject the patient's blood. We just inject this one tumor lymph node at the left inguinal site. And after the vaccination, you see that there's 42% of his blood that's taken up by tumor is now gone down to 6.6%. And we stopped the therapy. That's a 3-month time point post-vaccine. But if we look 3 months further down the road, now the tumor cells in the blood have continued to regress. Again, this is a hint to us of inducing anti-tumor adaptive immunity. This is looking at that data, B cells versus T cells, but we can look at the same data in even somewhat more elegant way if we look at the patient's healthy B cells versus his cancerous B cells. So this is really the same data, looking in his blood at these time points, but now we have on the x-axis lambda light chain. This patient's lymphoma is a kappa-expressing tumor. So again, 42% of these cells in his blood are his kappa-expressing B cells, which are his tumor cells. And we see there's a tiny number on the bottom left here again of healthy B cells. They are lambda-expressing healthy B cells, 0.4%. As we move along during the therapy, again, those tumor cells regress, but somehow the immune system is able to distinguish the cancerous B cells from the healthy B cells, and you see those healthy lambda-positive B cells increasing even while the cancerous B cells are decreasing from 0.4% to 1% to 1.4%, and even more so over time. So we hopefully have trained the immune system here to recognize cancerous B cells and distinguish them from healthy B cells. But the obvious question there would be, what is the tumor antigen that these T cells are reacting to? We don't have the answer to that question. We are actually working very aggressively to get that answer, but we don't have it yet today. With the FLT3 ligand therapy in these patients probably caused many changes in the immune repertoire, so we've become more thoughtful since starting the trial, and now look at all of these patients instead of with flow cytometry, with mass cytometry. So here's a mass cytometric plot, a SPADE plot, looking at the patient prior to FLT3L therapy and after FLT3L therapy. And I'll just show that transition one more time, focusing on these bottom few cell subsets like the dendritic cell subsets. Very few dendritic cells before, and about a 10-fold increase in both the CD141 dendritic cells, a very rare subset, and in the CD1C dendritic cells. I gave a hint of that with flow cytometry already, but now we can look at every subset, and when we look at every subset of cells, the entire immune repertoire, we discover things that we would not have even been thoughtful enough to ask about. And as an example of that, here's actually an even more honed-in mass cytometry panel focused on T-cell markers. And again, there are so many markers we could look at on T-cells. It would be hard for any of us to pick which combination of T-cell markers we should focus on to sort one group of patients versus another. But here, if we look at responding patients versus non-responding patients, and we ask the SPADE algorithm to just find us differences, we see here on the top panel that these responding patients have this group of T cells that I've circled. These are CD8 T cells that express high amounts of PD-1, LAG3, CD38, and TIGIT. So there are a reasonable number of those cells in all of the patients, the responding patients and otherwise. But the color code here is fold change in that T cell subset after therapy versus before therapy. So you see these top few patients have very little change in those T cells, in that T cell subset, whereas the non-responding patients have, again, this was a log scale, so this is a many-fold, I think it's about a 4 or 5-fold increase in the PD-1 high, LAG-3 high, et cetera, CD8 Tcells after therapy. And this is similar to something that other groups have discovered with other types of immunotherapy, that as the CD8 Tcells are learning how to attack the patient's cancer, that we have induction of adaptive resistance, meaning that the cancer tries to turn off those T cells, and these T cells start to express some of these checkpoint molecules, PD-1 being the most famous, but all of the others being important as well. I mean, this is not, just academic and interesting. This is very plainly actionable. If these are the anti-tumor T cells, and again, I haven't proven that here, but it's just a suggestion from the precedent of prior recent publications. If those are the anti-tumor T cells and they're expressing a lot of PD-1, simply adding an anti-PD-1 antibody to this therapy would be the obvious next step. And so that is our question, and just in these last few slides, I will discuss how we plan this for the future, both in our mouse patients and in our human patients. So this same therapy could be improved in a few ways. One way is that instead of stimulating those dendritic cells with a TLR agonist, we have other approaches. One famous example are oncolytic viruses, which can be immunostimulatory while they also kill some cancer cells. So we see here that as we add in this Newcastle disease virus, which is a focus of some of our collaborators here at Mount Sinai, that we have an actually profound effect by the addition of FLT3. So just cutting to the survival curve here on the right, we see that the Newcastle disease virus in the red curve does allow some mice to have prolonged survival, and a few mice are cured, maybe 10%. But doing that same therapy in the context of FLT3 ligand, when there are more dendritic cells in the tumor, now we cure about 80% of the mice. And this, again, is hopefully very translatable. Newcastle disease virus has already been studied in clinical trials. One of our hopes is to hopefully begin to incorporate FLT3 ligand with these oncolytic viruses. So perhaps the even lower hanging fruit in terms of clinical translation is something that's already being used every day for our patients in the oncology clinic, which is assessing the PD-L1 expression of these tumors. So there's a concept called adaptive resistance, whereby when we induce anti-tumor immunity with CD8 T cells, their expression of interferon gamma causes PD-L1 upregulation in the tumor itself. So in this same mouse model of the in situ vaccine, we see after this triple therapy, FLT3 ligand, XRT, and poly-IC injection, if we look at those tumors in the third histogram, we see that PD-L1 expression actually does increase probably about 5-fold as compared to baseline after the vaccine. So this is looking at all of the conditions, and only the triplet therapy causes increase in tumor expression of PD-L1. So not surprisingly, we have reassessed this maneuver using anti-PD-1 antibodies provided systemically. And delightfully, we see here on the bottom these tumor growth curves. At this bottom curve on the bottom left panel, the green curve shows that almost all of the tumors regress completely as compared to the black line control, or the red or blue lines, which are the in situ vaccine in the red line, and PD-1 plus radiotherapy in the blue line. Vaccine, again, looking over on the bottom right, the Kaplan-Meier curve, we see the vaccine cures probably about 40 or 50% of mice, but combining with anti-PD-1 antibodies, now we cure 90% of animals, and that was pretty gratifying to us, and also sufficiently promising that we now have been able to set up a deal with the Cancer Research Institute, who will fund a clinical trial later this year of exactly that approach, combining the in situ vaccine with anti-PD-1 antibodies, and again using very similar assessment of distant regressions to show that the therapy is working. And it's kind of nice that we'll also be able to get beyond lymphoma with this trial and also treat patients with breast cancer, head and neck cancer, and maybe other superficially accessible cancers. So combination with anti-PD-1 is probably the obvious thing in terms of clinical translation. PD-1 is by far the maybe greatest success story in all of cancer immunotherapy in the last 40 years or ever, but it doesn't mean that PD-1 is necessarily the best checkpoint for us to block. So to try to get at the question of what might be alternative or even better checkpoints, and by checkpoint again I mean molecules that anti-cancer CD8 T cells express that prevent their optimal anti-cancer function. We are using a novel mouse model called the Jedi Mouse. The Jedi Mouse I'll describe here in a second, and first I will delight in what a wonderful website Google.com is, because if you type in Jedi Mouse, you can find several great pictures. This was the best of all of them. So if you guys are not familiar with the website Google.com, I would definitely recommend it. I think we can put a link for how to find Google.com if you're not familiar with it. So the Jedi Mouse is this mouse that was developed by our colleagues in the lab of Brian Brown last year, published here in Nature Biotechnology. And this mouse generates CD8 T cells which recognize a peptide of the Gfp molecule, the green fluorescent protein molecule. So basically, these T cells can kill any cell that is green. This has a tremendous number of opportunities for application. I'm going to skip these videos here for the moment just to show you a later video of how that killing occurs. But basically, we can use the Jedi CD8 T cell to kill any green-expressing cell, specifically in the class 1 H2D background. And here's an animation of that. On the bottom right, we see these green cells melting away, while the top left red cells and the bottom left unfluoroprotein conjugated cells are intact. I'll just show that animation one more time. The right panel is just with more Jedi T cells, the left panel with no Gfp specific T cells. Let me show that animation where we see specific killing, especially here on the right at the high T cell level of green cells melting away. And at the end of 7 days here, you see there are very few or almost no Gfp expressing tumor cells left. So we're going to take advantage of that in the following way. We have developed a CRISPR library of 2,400 elements targeting 600 genes that are highly expressed in these lymphoma cells. So we have, as you see here on the left, red transduced, red fluoroprotein transduced lymphoma cells. On the right, green GFP transduced lymphoma cells. And just as a thought experiment, we'll say they all express a checkpoint molecule. We'll call it protein 3. Think of this as a PD-L1-like molecule. And T cells have some receptor for that PD-L1 molecule, a PD-1-like molecule. And when protein 3 hits receptor of 3, those T cells do not kill as well. Therefore, when we put this CRISPR library into all of the red and green lymphoma cells, only the lymphoma cells that get CRISPR number 3 will lose expression of protein number 3. If protein 3 is just a PD-L1-like molecule, not a survival molecule, the red cells will not be harmed at all by their lack of 3, whereas the green cells that lose 3 will no longer be able to inhibit anti-cancer T cells, anti-GFP T cells, and they will be selectively killed at a higher rate. Therefore, afterwards, all we have to do is sort the red and green lymphoma cells, sequence the CRISPRs in each, and look for every element that has a high red to green ratio. Anything with a high red to green ratio is a PD-L1-like molecule. And that is the concept, and here's us just trying out that concept, not in a library of 2,400 elements, but just in a library of one element, getting rid of PD-L1 as a poster child of these checkpoints. And what we did is we CRISPRed out PD-L1 from red and green cells. You see in the leftmost flow plot here, we've made a sort of a 40-40-10-10 mixture of left red, right green, and in the bottom two panels the same, but PD-L1 CRISPRed out. So we start these co-cultures as a 40-40-10-10 mixture. In the middle panel, you see when we add anti-GFP T cells. In the top two panels, we see now the Jedis are killing well enough that instead of a 40-40 ratio, they have a 64-9 ratio, so about a 7-fold killing, and that's in the normal conditions. But in the bottom panel, the same exact test tube, in the PD-L1 gene-edited out version, now instead of a 64-9 ratio, there's a 26-0.1 ratio, so something like overall a 185-fold killing instead of 7-fold killing. And so really the metric here is that ratio of ratios. 185 is about 27-fold better than 7. So the metric for PD-L1 killing is 27, meaning that without PD-L1, these T cells kill 27 times better, and we're going to get that metric for every member of this CRISPR library. And the library has been sort of thoughtfully annotated. Here is that library of 600 elements. These are things that are highly expressed in the lymphoma cells. are somewhat immunologically annotated, meaning they may have some role in immunomodulation, and they are specifically things that are either surface expressed, that we could target them afterwards with an antibody, or kinases or phosphatases, things that we could block with an inhibitor. And we are looking to bring that to patients, but we're working with a few different venture capital teams to see how we can get that going quickly into a translatable platform. And I'll mention one other way in which we're doing a very similar approach using the Jedi T-cells. We call this the reverse genetics approach as opposed to the forward genetics approach, meaning that here, instead of forcing a genotype with a CRISPR, instead we force a phenotype, the phenotype of T-cell resistance. And we force that phenotype by just, in these co-cultures, you see by day 4, we're already starting to kill most of these green lymphoma cells on the top left here. We take those surviving cells and we flow sort them for Gfp high cells and also class 1 high expressing cells, so that we don't just take the survivors, but we take the survivors that are not cheating by just getting rid of the Gfp transgene. We sort those and reco-culture them again. After 1 generation, we don't see much difference in their ability to resist T cells, but after 2, 3, 4, 5 generations, now we start to see some inability of T cells to kill these, again, iteratively selected Gfp expressing cells, such that in the 5th generation here, we call this R5, resistant generation 5, after 4 days of co-culture, those cells are not getting killed at all. Instead of 46 going down to 17, now 46 is staying at 46%. And that's the 5th generation. We actually have continued this up to the 8th generation. I'll show you a video of what that looks like. The top green cell here is the parental T-cell sensitive cancer cell. The bottom yellow cell also expresses green. In fact, it expresses just as much green, as much Gfp as the sensitive cell, but we've added another dye to it so it appears yellow so we can tell the difference between them. And all of these other cells around here, these are almost entirely Jedi CD8 Tcells, and you will see as we're going to go to the video that, and you will So you saw there that that top green cell, the T-cell sensitive green lymphoma cell on top, not only was it killed about 3/4 of the way into that video, but you saw the T-cells forming many immune synapses all around the cell, whereas on the bottom, this other cancer cell, really the T-cells almost seemed bored or sleepy while they were surrounding it. And that's not because the cells have different amounts of Gfp or different amount of class 1. You see these different cells have, in fact, the resistant cells have even a bit more Gfp because we keep selecting for high Gfp. And they have the same amount of class 1, and they happen to have the same amount of PDL1. So what is the difference between these T-cell resistant and T-cell sensitive cancers? We thought maybe it was just that the T-cell resistant cancer somehow changes the epitope so that T-cells do not see it at all. And you see here when T-cells, now we're looking here at Jedi T-cells, when they see the sensitive cancer, they start to upregulate within 96 hours activation markers like CD69 and CD25 here in the right panel now. And we thought that maybe when the T-cells saw the resistant tumor that would not happen. But it's exactly the opposite. Just as the same as with this T-cell sensitive tumor, when the T-cells see this resistant tumor, they still start to upregulate CD69 and CD25 by 96 hours. So what's the difference? The difference is that although they are expressing those early activation markers, when T-cells see the sensitive tumor, they start to also express cytokines, interferon gamma and TNF. But when they see the resistant tumor, they get completely shut down somehow. They do not express hardly any interferon gamma and only a very small amount of TNF. So again, the early activation is occurring, but something subsequently is turning off the ability of these T-cells to go through complete activation and tumoricidal function as expressed by interferon gamma production. So we performed RNA-seq on the sensitive versus resistant tumor cell lines, and we have a list of candidates of things that are increased here on the left, things that are increased on the resistant tumor cells. We also on the right have things that are decreased on the resistant cells. And we have moved forward to pick perhaps our favorite candidate so far, and I unfortunately can't go too much into the details of the specifics of each candidate, but this candidate, called candidate 33, as you can see here by a flow plot, is highly expressed on the resistant lymphoma cells. It's actually a bit bimodal, but you see in about 70% of the cells, they have about 20-fold more of this candidate protein on the surface than the parental sensitive cells. And that molecule is a transmembrane molecule involved in signaling. And so over here on the right-sided panel, you can see that the bottom two flow plots are demonstrating phospho-ERK, so signaling within those tumor cells. And that marker is something that ligates a phosphatase intracellularly, and not surprisingly results in hypophosphorylation of ERK. So that molecule is both highly expressed on the tumor cells and is highly functional, decreasing downstream signaling in the tumor cells, just to show that there is more of it and it's functional in the tumor cells. So we won't get more into that molecule. We're still validating that this is the resistance mechanism, but both of these approaches are ways that we hope to discover the next PD-L1-like molecules expressed by cancer cells, lymphoma cells, as well as other cancers. And I have to now most importantly acknowledge all the people that did this work, the members of my lab, my many collaborators here at the Mount Sinai School of Medicine, and very importantly our core facilities, especially our immune monitoring core led by Sunji Kim Schultz and Adib Rahman, that really have a veritably plug-and-play model set up so we can get mass cytometry, cytoph experiments done both on our patient samples and on our animal samples from the lab very quickly. And I have to really congratulate them because they were just awarded a grant to get a second cytoph machine. That's how busy our first cytoph has been, that the throughput is just about maxed out now, and we're lucky to have an opportunity now to get a second one, as well as a tissue cytoph machine, which is coming later on this year as well. And of course we have to thank the people that help fund the research, and we sometimes don't emphasize this enough, that we have to thank the patients that are involved in these clinical trials, because they are really going very well out of their way to both look into a novel therapy that may be effective or might not be, but also to go along with all of the extra requirements involved in us getting the samples needed so that we can try to learn something from these clinical trials, and hopefully I've expressed some of that learning here for you today. And I'll be happy to take any questions.
Excellent presentations, Dr. Poulin and Dr. Brody. Thanks for bringing that information to us. Before we get started with all of your questions, here's a quick reminder about how to reach us. Questions can be sent via the green Q&A button at the lower left of the presentation window. We'll get to as many as we can. Our first question is, do you have those Gfp A20 tumor cells commercially available? And this is for Dr. Brody.
Great question. You guys can hear me, I think. GFP fluoroprotein transduced A20 cells are not commercially available. They're fairly easy to make, and if somebody wants to contact me, we could probably figure something out. Good question.
Great. Another one for you. What did mass cytometry add to your analyses that flow cytometry would not have?
It's a great question. I actually will try to scan these slides over to slide number 27 that I think maybe are one great example of an answer. I mean, here we are looking at a T-cell focused, a CD8 T-cell focused mass cytometry panel where we have many markers, I think 434 markers, and we of course never be able to fit all of those into a flow cytometry panel, and wouldn't, if we had used 4 different panels, not known which markers to associate together. We might not have had PD-1, LAG3, CD38, and TIGIT all together, but with those markers all together, we found this particular subpopulation of what we believe are highly exhausted, possibly anti-cancer T cells that increase in the non-responding patients and stay the same in responding patients, don't increase in those patients. So we can go back by flow cytometry now. We think that flow cytometry is not dead. We can go back and use these surface markers to sort these populations and test them functionally, see if they really are exhausted cells. And of course, this can guide the next round of trials, which will be to add anti-PD-1 antibodies, and maybe in the future multiple antibodies as we move forwards. And that's a good question as well.
And for you again, how do you use immune monitoring data and or mouse study data to make decisions about immunotherapy choices?
That's a great question and a difficult one because that is really the problem of the field of immuno-oncology. We have what's a quote good problem to have. We have too many choices, but the problem therefore is we don't know how to combine all the possible choices. We have, just to pick a group of molecules, monoclonal antibodies against OX40, LAG3, TIM3, of course, PD-1 and CTLA4, and easily a dozen others plus small molecule kinase inhibitors, IDO inhibitors, and so forth in the clinic. What's the right combination of these things? We really don't know, and unfortunately it's driven a lot by a sort of a separate agenda, industry, pharma, and biotech agendas of what they happen to have on their shelves, which is okay, but not the ideal way to drive combinatorial strategies. So we really need to be looking in the trials that we do. We give a lot of lip service to biomarker studies, but we really need to be obtaining tumor samples during therapy to see what is the state of the T cells, ideally, as we're able to, the state of the anti-tumor T cells, as opposed to the rest of the non-specific T cells, to see what we could do to be helping those T cells kill cancer more effectively. So we can learn some of that from really in-depth immune monitoring of our ongoing clinical trials, but it really has to be done and has to be prioritized when we write up those clinical trial protocols.
And Dr. Brody, this one's for you. Did you test different types of PD-1 monoclonal antibodies other than Pembro when you considered addition of PD-1 blockade to your in situ vaccine approach?
Yeah, that's an interesting question. I'll tell you quite honestly, we think the majority of PD-1 and PD-L1 antibodies that are either FDA approved or in the clinic are probably more similar than different. So those decisions probably get made more at a practical level of which industry collaborators are looking for this type of collaboration. So yeah, we are open to pembrolizumab, nivolumab, atezolizumab, all of the others that are probably going to be FDA approved soon. And in different trials we use quite literally we have different trials, some with nivolumab, some with pembro, some with atezo. So we are open to all of this.
And Dr. Brody, can the activated anti-tumor CD8 Tcell be found in patient peripheral blood after treatment?
They definitely can be. It may not be the best place to find them, and depending on the timing and what the therapy is. So, for example, after any anti-PD-1 therapy, we think the chances are much higher because we sometimes find them in the peripheral blood after anti-PD-1 therapy, and then we go back and sometimes look with tetramers to see actually if there was a small proportion even prior to therapy, and there frequently is, but it's a very small number, and it's hard to find the signal versus the noise. So yes, we can find them in the peripheral blood. Again, sometimes they might be in the blood, and that may not correlate with anti-cancer effect if the job, of course, of those T cells is to get to the tumor site. Just generating them peripherally might not be enough to induce anti-tumor effect. We need to see them at the tumor as well, but we can find them in the blood, which obviously is much more accessible.
Dr. Brody, how do you decide the frequency of tissue monitoring post-therapy?
It's a somewhat practical question as well. Some of these therapies that I talked about in situ vaccinations rely on our ability to access the tumors literally, with a syringe and a needle. So they are superficial tumors, things like lymphoma, breast cancer, head and neck cancer, and therefore we can do, fine needle aspirates or core biopsy of these. It's not that fun for patients, and we do these every couple of weeks, and patients can tolerate that. If we did them every day, patients would not tolerate that. And similarly with peripheral blood sampling, if we do this every week or two, patients tolerate that. Much more than that is difficult, not just because patients don't like getting poked with needles, but because these patients live somewhere between 30 minutes and 12 hours away. Some of these patients are traveling cross-country to be part of these trials. So it's just not practical to do too much monitoring. Of course, the expense and the resources involved are a practical concern as well. But we try to do weekly monitoring in the near term after the start and during therapies, so we have a sense of how they are changing the immune repertoire. Sorry about that. I just had my speaker go defunct. If you could repeat the last question.
So, Dr. Brody, I think we have time for one more question. The question is, FLT3 ligand is known to expand many other myeloid cell types, including monocytes and myeloid-derived suppressor cells. How would you overcome the possibility of potentially inducing some of these other cell types?
Yeah, it's a great question, and while you're asking it, I'll just flip back to these slides, number 25 and 26. This is prior to FLT3 therapy, CyTOF spate analysis, and here's after FLT3 therapy in the peripheral blood of one of our patients. And as the questioner asks, we do see these dendritic cells populations increasing many-fold, but we do see these other shifts, these blue to green changes in the, we'll just generally call them myeloid compartments. Some of them are CD14 monocytes, some of them are other types of myeloid cells. And some of those could be MDSCs. Really, I think in terms of MDSCs, another related differentiation factor, GM-CSF, is the most famous for inducing MDSCs. But it's possible that FLT3L could also induce some tolerogenic myeloid cells. You know, one way we approach this is we definitely do not think FLT3 ligand therapy would be a good monotherapy. Quite honestly, I think it would be more tolerogenic than immunogenic. So an important part of our approach here is that we need to activate those myeloid cells after they're recruited, and that's with the application or intratumoral administration of toll-like receptor agonist, be it poly-IC-LC like in this trial, or other approaches, other TLRa agonists, other STING agonists that we're using in the clinic now. And those can sometimes also even convert MDSCs to more functional M1 type macrophages or other probably helpful inflammatory myeloid cells instead of the tolerogenic myeloid cells. So I think the answer is these cell recruiting factors are not a standalone therapy. We need myeloid cell activating factors concurrently, be it TLR agonist, STING agonist, or even perhaps oncolytic viruses. That's a great question.
Thank you. I'd like to thank the audience for all their great questions, and if we didn't get to yours, we will follow up with you via email. I'd also like to once again thank Dr. Michelle Poulin and Dr. Joshua Brody for their presentations. Do you have any final comments?
I'd just like to thank everyone for the great opportunity of sharing some of our work.
Thank you. I'd also like to thank our sponsor, Fluidigm, for making it possible to bring this presentation to you. Today's webcast will be available for on-demand viewing through October 11th, 2017. You'll receive an email from us alerting you when it's available on demand and posted on LabRoots.com. You are welcome to forward this announcement to any colleagues who weren't able to join in today. Thanks for logging on and participating in today's broadcast. We'll see you next time.